I'm not going to catch my breath, so we might as well just start. Is there any way to make this smaller? To go to meeting stuff? No. Whatever, just put it as that. Sure, so let's start. If you start the recording. All right, just go ahead. Just a quick introduction. I'm Patrik Bay. As all the previous presenters in the summer school, I also work in the brain simulation section with Petra Ritter. And today I'm gonna present a little bit as a second to last talk now, a processing pipeline that we developed. It's called Lesion to TVB, that has the goal of creating TVB-ready data for stroke patients, for stroke patient populations. So the idea is a little bit, to give a quick overview. There you go. To give a quick overview of processing of such multimodal neuroimaging data in general, then go quickly about what are the requirements and what is the processing necessary to actually do TVB, what are the specifics and the specific challenges when we're talking about stroke. Then I'm going to present the pipeline that we developed and implemented on a high level and go into a few details on some of the major aspects of it. Present the extensive validation that we did for the pipeline as a whole, but also for some of the minor adjustments that were necessary. And at the end, showcase how we can use this pipeline to not just run any TVB simulations, but to really try and focus on how do we get stroke data all the way to TVB simulations and how we can use TVB to study the mechanics and potential strategies towards recovery when we're talking about stroke, which is the ultimate goal, to have that clinical relevance of our simulations, right? So the idea is, and I guess you've seen this now during the talk, how do we get from the initial subject, or in our case, actual patient in the scanner, to our brain model, to our TVB model, while acquiring different modalities, different types of neuroimaging data, right? We're talking about anatomical data, track data that really capture the connections between the different regions, and actually creating this overview of the different regions of the brain, this parcellation with the regions of interest, to then create a model that not only represents the regions accurately, but also represents the connectivity between those regions. So the strength of the communication pathways between the regions accurately, and then use this in TVB with mathematical modeling to create the model and actually have our nice simulations that we can see here. So the basic neuroimaging data, if we start with this, is the anatomical data. Typically a T2 and a T1 image that really captures just the different tissue types of the brain across different sensitivities of the acquisition technique. You can see this already if you look at the raw data here, always on the left. We have the skull in bright white. We have the white matter, which is somewhat gray. We have the gray matter, which is the darker gray, represented in our data. We have this for the T2, which is in our case actually a fluid attenuated inverse relaxation T2, a sort of flare image, which puts a little bit of focus in the acquisition technique on capturing liquids, one could say, in the brain. So it's relatively common in stroke patients because it really helps the radiologist to perform diagnosis and segmentation of the actual lesion, because it highlights it very well. So the idea is that we start with these raw image data and we end up with the fully processed structural data here. Actually, I cannot see this exactly, with the structural data here that we can then use to also create this parcellation that we have, as the ultimate goal is to have the different brain regions being represented in the subject space. So for each individual subject, perfectly following the anatomy of the subject, representing in volume of the region, location of the region, that we can have individualized brain models at the end. The other, switch again. The other main data acquisition technique that we have is the diffusion weighted imaging, where we go, where the focus in the acquisition, the physics actually of the acquisition has a little bit of a different focus. Now we're trying to capture, instead of the actual intensity at the voxel, we're trying to capture and then extract the pathways of the brain. So here we're talking about the white matter tracks that we want to extract to get these nice little colorful images of the tracks that are actually going through the brain. So we're starting from the raw image here, performing a lot of pre-processing, normalization, bias correction, distortion correction, all these artifacts in the data that are where there's really no way around it. This is just because of the physics of the scanner that we have this introduced into our data, and we need to take care of this. Get to this a little bit later as well. And we want to have these nice little tracked images of the brain so that we can then use the parcellation that we had before, right? We have the individual regions of the brain so that we can go through all of the regions and really pretty much just count the number of tracks from region 1 to region 2 so that we can end up with the structural connectome where we have all the regions in our parcellation. In our case, this will be a total of 383 regions, which is a combination of the 360 region of the Human Connectome project, I get to again as well later, and 23 subcortical regions that we included to really capture the full brain in our data. And we see a lot of interhemispheric connectivity here in the left hemisphere, a lot in the right hemisphere, a little bit cross-hemispheric connection, which you would assume and also expect, and a lot of them here, everything above the 360 to 383, a lot of subcortical connection as well. So we can really nicely capture the individual structure of the subject. In a similar way, we go for our fMRI data, where we start again with the raw image data suffering from a lot of those drawbacks that I mentioned. For example, we can clearly see a distortion here, which is introduced in the frontal cortex here, just by the direction of the acquisition. You can imagine it going pretty much from front to back, applying the magnetic field to our data, and then we have a lot of distortion here in the beginning, and we're trying to mathematically correct for these so that we end up with a nice representation of the whole brain for our fMRI data and normalize so that we can see we're actually having this higher activity along the cortex rather than in the white matter that we have here in the raw image. And then based on this processed image, we can do the same thing again. Take our parcellation, take our regions, compute the average activity for each region that we have, and then instead of having the tracks that we have, now we're looking at temporal connectivity, temporal correlation. So we compute the Pearson correlation coefficient between the average time series for all the regions, and we end up with our functional connectome. Again, the same structure on the left, the right hemisphere, and the cross-hemispheric correlation as well. And this is pretty much just the basic idea of what we want to do, what we want to end up with in terms of processing. Now, because we're talking about processing pipelines, we always want to take a look at the gold standard, and really the gold standard for processing these multimodal neuroimaging data, and at least these three modalities that I've mentioned before, the anatomical, the functional MRI, and also the diffusion imaging. The gold standard currently is the minimal processing pipeline from the Human Connectome project. It's a GLASA 2013, and in addition to this, a 2016 paper, where we can really see as the final result where we want to end up. Right? We have a functional MRI image, which is the pixel image down here, and the extracted activity in some region of the peripheral occipital cortex back here, and we have overlaid in green and in blue the gray matter, white matter barrier. So the cortical ribbon we extracted, or they extracted from their anatomical data, overlaid to the functional data, and really nicely showing the high resolution of the cortex that they were able to extract, and the almost perfect overlay of those regions and the regional activity that they extracted with these cortical ribbons. This is one of the first instances where we can already see a little bit of an issue when we're talking about this kind of processing, It's just the sheer difference in resolution and in properties of the acquisition technique itself. Even though the HCP data is of the highest standard, and we're talking about sub-millimeter resolution for the structural data, we still have the functional data being rather pixelated, as we can see here. Right? So we see the overlap of the cortical ribbon and the voxel actually not being a perfect match simply because of the difference in resolution of the acquisition technique. So there's a lot that we have to control for if we want to actually have an accurate representation of our functional, but also of our structural properties that we're trying to capture. This just as an example for where we want to go and what we would like to reach also with our pipelines for the stroke patient, because the human connectome project only covers healthy subjects in that sense, at least when it comes to pathologies or large artifacts in the brain. This pipeline is not able to handle it. So to take a little bit of a closer look, what are those just basic processing steps that we really want to perform and that are continuously performed throughout these large pipelines. Right? We're talking about pipelines running for 20, 30 hours, but they continuously go back. And if you look at the actual tasks that are performed, these are some of the main basic tasks that we want to look at. The first one being just registration, either cross-modal registration. So we've seen the different modalities. We want to have a diffusion image, right? We want to have our tracts and our final pastillation in the same space. One is created in the T1, the other one is in the diffusion space. Even though the subject is the same subject and was in the scanner at the same time, one acquisition after another, there's still movement, there's still a lot of aspects that actually change the space in which those are acquired, as well as the actual resolution of the space. So there's a lot of registration needed across the different modalities to make them overlap. Same goes if you want to register to a template space, such as, for example, the MNI template, to make different data types and different studies, for example, really comparable based on a common template. We're talking about surface reconstruction. I think we've seen this in one of the previous sessions as well, where in TBB we can also run surface simulations. So having an accurate representation of the surface of the subject is really helpful. And surface reconstruction is one of the main and most computationally expensive processing tasks that we want to perform. The next one is segmentation itself, where we want to have the different tissue types, for example, in our T1 image, gray matter, white matter, cerebrospinal fluid, etc., segmented from the different image that we can then use as the base for our pastillation. Motion correction is one of the main aspects that always have to be performed. We're talking, for example, functional MRI imaging, because now we don't just have a single image being acquired, a single 3D image, but we end up with a 4D time series. And if we are talking about task-specific data, we have the subject being in the scanner for a lot of time, and even to see the resolutions becomes smaller and smaller, 2 millimeter, 1 millimeter, even in functional imaging. Just slight head movements have a lot of impact on the actual positioning of the brain and of the single voxel. So performing motion correction, there is a lot of a very important aspect. There are different techniques for this, and I have to go through this into too much detail. And the last one is rather vague, is the artifact correction with a typo, which is especially important in our case when we're talking about stroke, because such a stroke lesion pathology can represent one of the largest really physical artifacts that we can have in our data, and we have to make sure that we accurately and robustly take care of this to enable the actual processing. So what is the idea of registration? Right? We want to have, if you're talking about, for example, T1 and T2 images, we want to have them overlapping in the same space, the same voxel being at the same position across the different modalities to really, for example, in the case of the HCP pipeline, we can use the gradient between the T1 and the T2 image to indirectly estimate myelin maps of the brain. So we really want the voxel in the T2 to be the exact same position as in the T1. So we're trying to register the images to each other, which basically just means minimizing the distance between the same voxel in the T1 and the T2 image. There's a cost function that is being applied. there are different versions for this, depending on whether or not we have the same representation of intensity values. For example, this is one of the differences in T1 and T2. We see the change from the gray matter being a dark gray to the white matter being rather a light gray, as opposed to the flare image where the gray matter is represented as white, and the white matter is actually turning more into a gray and to a darker gray. So if you're trying to, just based on the intensity values, create a minimum distance here, we have to apply, or we have to be careful of the cost function that we apply, as just an Euler distance, for example, is just a norm between two intensity values will give us the wrong warping, because it's trying to map a very bright voxel to a very dark voxel. So there are different aspects that can be how this can be avoided, and we want to end up with the perfect overlap. We're seeing here in gray the T1, and in gold overlapping perfectly the T2. Just cut out a little bit of the T1 visually here to really show the perfect match between the T1 and the T2 image. Even though this is actually a flare and there's a lot of difference in resolution, we end up with this really nice overlay if we perform the correct and careful registration that we want to have. Like I mentioned before, the segmentation that we want to have, we're starting with the T1 image. Actually, we're doing this on a rather processed, so we already have a skull script, have the brain extracted, etc. And then we want to actually extract the different tissue types. Here, the most important, right, the cortical ribbon, the gray matter that we want to have, the white matter representing the tracks, combining that, but also the cerebrospinal fluid really encapsulating the whole brain and filling those spots. There are different tissue types that can be computed. Another one that is also important in our case, that we'll get to a little bit later as well, is the pathological tissue, which is just a category to include any types of pathology that you want to define, even if they are represented in different intensity values and different properties, etc. This is one way to try to segment the lesion inside of the actual T1 images, which is an ongoing research field, and currently, unfortunately, there's no real way around the radiologist or neuroradiologist actually looking at such a data and manually segmenting the lesion. So also in our case, this is required. We have to manually add this pathological tissue type, because automated segmentation algorithms, currently a lot of them are using deep learning algorithms, do not fulfill quality requirements if we're talking about a clinical context. So this is an ongoing, very active research field. And then as another one, we want to have the surface extraction, as we discussed this. And why do we want to do this? Why is it actually a more accurate representation of the individual topology that we have in the brain? It's easy to understand if we think what the data represents, right? If we're talking about the volume of volume image, we're talking about 3D cubes, those pixels representing the intensity at that spot, as opposed to the surface mesh, where we're talking about a triangular representation of the actual surface of the fat. And what this allows us is to really circumvent one of the major disadvantages of this volume space, where if we're talking about, for example, getting the distance between two regions or getting the distance between two just voxels, in that case, if you're thinking about having the one voxel represented on this side and the other voxel represented on the other side of this cortical folding, if you're just taking the difference in coordinate space of this 3D voxel, it is relatively small, underestimating the actual difference on the surface. And if we take the surface way and we're talking about the difference between the different triangles that represent the region, we see that we are actually going on the 2D surface along this cortical folding, ending up with a much larger distance, which more accurately represents the actual distance in the topology of the brain between the two regions, instead of just this three-dimensional distance in 3D coordinate space. And with those as the just basic building blocks of such pipelines, just to give a little bit of an example of how the HCP processing pipeline is built and what it is performing. It's separated into three main blocks, which is the structural pipeline, which is at the top here. It's the functional pipeline consisting of the fMRI volume and fMRI surface pipeline, and also the fusion pipeline. We're just looking at the structural and the fMRI volume here because those are the pipelines that we discuss in terms of our pipeline as well. fMRI, surface, and diffusion weighted imaging do not play into what we're actually doing with our stroke pipeline. So just to give a rough idea of what is happening, we're starting with our original T1 and T2 images with the corresponding field maps, which are actually not even necessary if you're talking about the minimum requirements. And we're going through step by step by performing distortion correction. So we've seen this before, especially in the EPI case, but also in structural acquisitions such as T1. We have those scanner-induced distortions and biases in our data that we're trying to get rid of. We're aligning here, another word for the registration, the T1 and the T2 image to actually get this perfect overlay. We're aligning it to some local landmarks of the brain for them to all have the same orientation in the brain. We're extracting the brain, performing again distortion correction, and ending up with the final T1 space that is somewhat as the goal for all other acquisitions that we have for all other modalities to be in that same space, because it's based on the highest resolution data that we have available, which is the T1 image. So we're having the most accurate representation of the actual topology of the individual. So all of the other processes are aiming at processing the corresponding modality into the same T1 space for the specific subject. A lot of this is a very simplified version or representation here. A lot of this is happening iteratively. So even, for example, if we're looking at the brain extraction, this is happening iteratively. There's an initial brain mass based on an MNI template. And then while we're going through even the surface extraction, we're taking more and more information that we have about the brain. And if we're thinking about, we already extracted the surface, then we know exactly where the brain is and what is CSF and what is skull, so we can update our initial brain extraction, etc., so that we end up with the highest quality process data that we want to have in the end. So we're starting here with the pre-free surfer part of the structural pipeline, then the free surfer, which is the surface extraction itself, based on free surface recon all algorithm, again performed multiple times. So even on an HPC environment, if we're talking about, for example, a 20-core call to this, we can still end up with 15 to 20 hour just pure processing time for a single subject. And then at the end, we have the post-first free surfer step, which kind of cleans up a lot of the interim results from the free surfer and also maps a lot of this to the HPC specific gray ordinate space, which we're not going to get into today, but at the end, it's also trying to incorporate the information that we had extracted during the free surfer to our initial pre-free surfer data. So we end up with our final extracted surface, with our final processed T1 image, etc. In the next step, what we're doing is the fMRI volume data follows a similar approach. Again, performing distortion corrections here as well, using the field map, while here is actually required to perform distortion corrections and even a motion correction to the fMRI image. And in the end, again, we're trying to register everything to the T1 image so that we're all living in the same space. As we've seen before, if we have our parcellation in T1 space, we want to have our fMRI and even also our diffusion weighted imaging in the same space so we can easily apply the parcellation that we have for our individual subject. So going back to TVB, what does this mean? What do we need to run the minimum, or what is the minimum requirement to run such a simulation? So we have our diffusion weighted imaging. If we want to process our own data, we require diffusion weighted imaging. We require the anatomical data, at least the T1 image, to get some form of segmentation and then parcellation, and bold images, fMRI data, to combine with the parcellation and get our functional connectome at the end, so that we can have our individual subject model based on the structural properties that we extracted, based on the diffusion weighted imaging, right? The tracks that we counted for the regions that we extracted, and then run a simulation and really validate it against the functional connectivity that we have available, because we want to map our model to really fit the data and create the functional connectivity and represent the same side of activity, so we can use our TVB model to try to dig deeper and use it as our mathematical magnifying glass, right? Which is the goal with TVB. That being said, about what we want to process, and if we have a nice healthy brain and high quality data, all of this is no problem. We have the HCP pipeline. Now talking about stroke, why is this so important that we actually did a project like this, and why there's so much research going on in this field? Because I mean, everybody knows that stroke, ischemic stroke, is still one of the major causes for disability worldwide, and it is a leading cause of death. So there's a huge societal toll and cost that this disease is forcing upon us. And still, we do still not have a clear correlate, even for the severity of a stroke, or in even better case, the recovery of the patient. Based on classical studies that looked at lesion volume, lesion location, etc., there's no clear mapping on whether or not the patient will recover, what kind of symptoms the disease will manifest, etc. So in the end, this sums it up pretty nicely, that effective therapies to support motor function after stroke are yet to be developed. And this is a 2019 paper, and we're not really a lot further than this now. So the question is really, how do we get to correlate the data that we have, which is representing the stroke, with the mechanistics underpinning of what is actually happening, why is this motor deficit manifesting, etc. And TVB here being one of the main drivers that we hope to establish to really get this research field forward. But what is if this is so pressing, this is such a good field to study, what are the main hurdles for stroke research? Why can this not be easily overcome? I summarize this at pretty much 3 categories that make stroke research so problematic in terms of the neuroimaging processing and the data itself. First one is just data. If we're talking about HCP data, those are high quality, healthy individuals that had a lot of time getting scanned, doing all kinds of tasks, and resulting in high quality data. In the case of a stroke patient, we're talking about a clinical context, especially if we're talking about acute stroke patients, which means just a couple days after onset, we are always in the clinical context, not within a research institute where the patient presents itself to be taking part in a study, right? We're talking about the clinical context. What this means is that the clinical context severely limits the availability of the data that we can acquire, and also the quality, simply because the main objective in getting MRI data for stroke patients is mainly from the radiologist to get a good diagnosis, to identify the location, to identify the type of stroke, etc. So the focus is not necessarily of creating data that can then later on be used for nice TVB or nice analysis studies with high quality processing. The next category is the disease itself and how heterogeneous the pathologies actually are. So even patients that might represent with the same or similar symptoms might have vastly different pathologies of the actual stroke. This includes location, but also just severity, volume, everything. So there is a lot of variance even in the patient population that is representing with this very similar symptoms. And the last step, methods, is just that because of the physics of acquiring this neuroimaging data and how a lesion might impair and affect this, we have that these artifacts can significantly impair performance and cause a lot of automated processing pipelines, such as the HCP, just to fail. In the best case, to inaccurately represent the data, but in the worst case, to fail. So you might, even though you might be able to apply such pipelines to some of your patient data, you might end up having a dropout rate simply because of the algorithm not being able to handle the large artifact of the lesion. So how does this look like if we're actually looking at the data? Why is this such a problem if we're talking about the clinical context and acute versus subacute phases, and even chronic? Because if we're talking about stroke studies, most of them focus on chronic stroke patients, which means the patient has recovered well enough, hopefully, and they're able to partake in such a study that is actually acquiring more high quality data, because now we are not in the clinical context necessarily anymore. So we're having just patients represented with the chronic stroke. the question is, why is this so different, and why is it so important to also capture acute strokes? And this is if we compare the data that we have here, which is the acute phase. In our study, that was 3 days post-onset, as opposed to the first longitudinal data set, which is still subacute phase, which is 2 or 3 weeks later. We already can see a significant change, and we can see really the significant dynamic of the lesion in the brain, right? Like with pretty much any injury that we have to our body, the body is amazing and adapts and tries to compensate. And here we see this large dark spot in the cortical, also in the subcortical regions representing our lesion. And then in the subacute phase in T2, we see that this has been significantly shrunk. We still have the lesion that we see, this white spot and a little bit of dark around it, that we can also find here. But even though we're talking about the same subject, we're talking just about a few weeks later, the image significantly changed. So if we're talking about processing this data and then, for example, extracting how specific regions of the brain are actually affected, we see something like the plot down here, where we have all the regions plotted, left hemisphere, right hemisphere, and subcortical areas. And what we have plotted here is the percentage of voxels of the specific region of interest that is covered and encapsulated by the lesion mask. So pretty much if a voxel is inside here, this dark spot, and the whole region is in here, the lesion load is 1. If the region is somewhere over here and is not directly affected by the lesion load is 0, which we see, for example, the left hemisphere here. And what we see the different colors represent being the time points, from time point 1 in the acute phase, time point 2, which is the blue, in the subacute phase, going to time point 3 and 4, which represent the chronic phases. And we can already see this dynamic really impacting the results that we have in the processing. Well, in the acute phase, we had a lot of regions even having a lesion load close to 1, meaning that it's almost entirely encapsulated by the lesion mass. Those almost entirely disappear even in time point 2 and 3. So the dynamic, the change of the lesion itself from acute to subacute and chronic significantly changes the inference that we can have about the lesion, and henceforth really impacting our ability to correlate this with behavioral data, with recovery, etc. We only see really a continuous impact in the subcortical areas, which kind of makes sense if we look at where the lesion now is and how this is actually represented. So we're just talking about subcortical areas here being affected, and it pretty much retrieved all the way from the cortical areas. The other aspect, if we're talking about the clinical context and the data, it's not just the disease in representation and manifestation in the data itself, but also, as I mentioned, the quality and the acquisition. This is most prominent if we compare again the gold standard, the HCP, which we have here at the top, and the example of the T2 weighted images, where we see still a high resolution T2 image, again overlaying with this subcortical cortical ribbon barriers that they extracted, seeing a nice resolution, very nice representation of the cortical ribbon and of the segmentation. When we now look at the FLAIR T2 image that we have in our data, we see already a significant decrease in resolution, and therefore in just the quality and the information that is contained in this data. We've seen this before in the T2 image. In the axial resolution, we have a high resolution, and this can really be used for the radiologist to segment the lesion. But if we're talking about sagittal and coronal axis of the scan, we're talking about maybe, in our case, it was 25, between 15 and 30 usually slices that we have. So you have very low resolution as compared to the T2 image, where we're talking about, I think it's 218 slices actually, even along the axial resolution. So we see a lot of difference here. The same, of course, is true if we're talking about the functional MRI data. Here again, the HCP, nice, covering the whole brain, it's covering high resolution. You can even in the fMRI data see the cortical foldings and the topology of the brain really nicely. If we compare this with the data that we have available in our data set, not only do we see a significant reduction again in the resolution, but we also see that this is so much smaller, not because I cut it off, but actually because the field of view in the acquisition of the data was significantly reduced. For everybody that has been doing MRI scans, and it can take a lot of time to really adjust the field of view to capture the whole brain for each subject. Again, time that you might have in a research environment, but in the clinical context, this might lead to data where you have data that might not be relevant in the direct diagnosis or treatment of the patient, as is in our case. So we end up with data like here, where we even have parts of the visual cortex at the end being cut off. We see this here, a lot of the temporal cortex missing. So the data quality is significantly impaired, to the point that it's actually a lot of missing data. So even the regions that that are not affected by the lesion itself, which represents a challenge for the processing, also there's actually missing data. So processing missing parts is obviously significantly different than to when we have a full brain available that we can use for our processing. The next category that we looked at again is disease and how the heterogeneous manifestation of these disease can be. And just as an example for our data, for our study, the study inclusion criteria during acquisition that was applied was it has to be a first ever ischemic stroke causing a motor deficit involving the hand function. So we're already talking about a very specific symptom that we have. It's very common in terms of stroke, but it's also very limited manifestation of symptoms that we included in the study. There's no aphasia, no hemanopia. All of this was excluded to really limit the actual symptoms that we have present in our population. But if we compare again these plots that we had before of 4 different subjects randomly selected, we see already the huge variance in representation of the lesions. We can see here a lot of regions being affected, representing large lesion volume on the right hemisphere in the subcortical area, just subcortical and wide one cortical area here, left hemisphere subcortical areas in this patient. And still, even though we have this variance in the manifestation of the topology of the lesion, we're already all talking about the same symptom. So the patient population that we're talking about here in the stroke studies is usually very heterogeneous, even in the manifestation of the disease itself. And in the last point, why is this so problematic for the methods? Well, most processing algorithms, at least independently, rely on intensity values and the corresponding gradients. We see this really nicely here in this example from the HCP, minimum processing pipeline paper, in the context of bias correction, to really reduce the bias, again, scanner-induced bias in our data to make the gradients of the intensity values more distinct. So what we can see is before bias correction, the segmentation of gray matter and white matter that we had is not perfectly accurately representing what we actually see in the image. And after bias correction, we see a nice alignment of the cortical rhythm extraction actually with the white matter that we have, really showcasing the impact of just slight adjustment to the intensity values and the resulting accuracy that we have. So why is this problematic in stroke? This is, I mean, if we just look at the intensity values that we have, this is already a very clear picture. If we're talking about the healthy intensity values, that's the yellow distribution here. And what we see on the x-axis is intensity values from very dark to very bright, and the frequency and how much they occurred. And what we did is here we extracted the lesion location and the intensity values inside of the lesion mask for the actual lesion affected hemisphere, and then we extracted the same area of the contra-lesional hemisphere. So we mirrored the image and extracted the same area again, and then we computed those histograms, and we can see that for the healthy tissue signal, we can see a clear bias towards the lighter intensity values, which makes sense if we just think about where we find these lesions. There's a lot of white matter in the brain, significantly less gray matter usually. So we have this strong bias towards the higher intensity values, so the brighter images in the T1 images, as we have this extracted from T1. And then if we compare this to the lesion distribution, which is very flat and very broad, we can instantly see that those are significantly different distributions. So now if we talk about algorithms like these, trying to segment based on the small gradients that we have between the gray matter and the white matter intensity values, a more smooth and broader distribution will mean that the gradient is less pronounced and therefore more difficult to extract the actual differentiation between gray matter and white matter, for example. So this is just at the core of all the impact that these different and abnormalities and intensity values can have for the actual processing algorithms. Going back to stroke and TVB, so we saw this what we needed, right? We needed diffusion imaging, anatomical and bold imaging to create our data to really run the modulations. And what do we end up with? Well, we end up with Q1 images, with structural images that have significant lesions, significant artifacts impacting the performance or even success of algorithms. We have low resolution modalities, we have the data quality being impacted. We have limited data even available in the fMRI, even to compute functional connectivities. And yet we want to be able to really build TVB models accurately. And for this, we looked at different approaches that can be deployed to really tackle these different hurdles. Again, based on the three categories that we have. For the disease manifestation, what is really been done? I mean, we are somewhat limited into what we can do there just because of the disease itself, and this is what we want to study, this is the goal. But just by restricting the patient population, for example, to specific pathological expressions, like, for example, people used to study or only include patients in the study with an identified lesion in the motor cortex, for example. That's the classic approach to those lesion studies. Another approach is to rather look at the network aspect, really ignore or not pay too much attention on the actual location of the lesion, but rather look how this is integrated into the larger network of those tracts, based on a normalized connectivity matrix, for example, just to tackle these different and various patterns in the actual disease manifestation. Which is not too relevant for us. For us, the main part is to look at the processing of the data. So how can we make this data that we have, how can we process this in a way that it is still robust and accurate? There are three main parts to this. First one being cost function masking, which is the technique applied to registration, which, as I mentioned, is at the core of a lot of different algorithms. It happens repeatedly throughout those pipelines. There is virtual brain transplant to just even enable the success of such algorithms, like, for example, FreeSurfer, to just be able to extract the surface if we have a huge hole or a huge lesion that is actually impacting the cortex. We see this failing a lot, so we're using virtual brain transplant to just be able to extract the surface at all. And then pathological tissue tractography, we go through all of those in a little bit more detail in a second, which is just a way of really trying to manage the abnormalities if we're talking about these nice little tracts that we want to extract, right, along the white matter tracts. How do we integrate the pathology there, and how do we treat it if we're trying to extract these traces? And the other one is, even though we have a lot of different stroke data available across different centers, and people do a lot of manual adjustments, etc. This really impacts reproducibility and just comparability between the different studies and the results. And containerization is one of the main aspects to really tackle this challenge, by just making a container available to everybody so they can use the exact same processing procedure to their data and make the results comparable. So those are pretty much the four aspects that we looked at. And the cost function masking, the basic idea is, I mentioned before, that if you want to register two images, whether that be to a template space, whether this be to across modalities, what we're trying to do is we're trying to reduce the distance, which can be seen as a cost function that we apply between the two images. So if we're thinking about, for example, we have subject data, patient data here with a large strokes lesion, and we want to register this to MNI, to a template space. The normal procedure here would be for the algorithm to pretty much go through all the voxels and try to minimize the cost function that we apply. Right? This could be cross correlation, mutual information, whatever. There are a lot of different cost functions that can be applied here. And if we want to do this, it's going to try to map this very dark region that we have here into healthy brain in the template space. So because in the template space, in the MNI space, we don't have this lesion present. So it's trying to really warp all the little bit of light intensity values that it finds to the brain. So what we end up with if we register to MNI space just normally is something like this, where it's trying to warp this region into the healthy brain of the template, and significantly impacts how the lesion is now represented. We can see, as compared to the original space, it looks nothing like the lesion that we had. So everything that we would do downstream out of this registration step is not anymore accurately representing how the lesion actually manifested in this brain. So the idea is to register or to reduce the optimization problem to only the healthy tissue. So what we're saying is, instead of trying to map the whole brain into the template space, map everything except for the lesion. So we're applying this mask of the lesion to the minimization problem, to the cost function that we're trying to minimize. So this is the optimization problem. The optimal warp that is then being computed is based only on the healthy tissue of the input image or the template image. There are different implementations of how this can be done. And then afterwards, this warp is applied to all the voxels. So now if we look at the data also registered to MNI space after the masking, we see that this is significantly better than without the mask, and is almost perfectly representing the lesion that we have in the actual subject space doing acquisition of the T1. So this enables us to perform registration without the drawback of having of losing the information about the actual lesion and how they are represented. And even sometimes, if we're talking about large lesions, this can also just fail because there's not enough overlap between the intensity distribution that it finds in the patient brain as compared to the template brain. The other aspect was the virtual brain transplant, and the idea is here to perform a virtual brain transplant. It's based on the idea of enantiomorphic normalization, which is pretty much built upon the idea that the brain is somewhat symmetric. We know this is not 100% accurate, but we know that the degree of symmetry between the hemispheres is sufficiently high for this to be somewhat accurate. So the idea is that to enable our processing algorithms, for example, surface extraction, to actually run, we're replacing the actual lesion signal that we have with the contra hemispheric signal that we can extract. So we're replacing this signal with the other hemisphere and ending up with something that looks like a healthy brain. Now we can actually perform surface registration because we see clearly here the cortical folding again, as opposed to here where we did not see it because we couldn't differentiate between actual lesion signal and cortical ribbon simply because of the similarity of the intensity values. If we remember, we now have this flat, very almost normal distribution of intensity values as opposed to the bias towards the lighter in the healthy signal tissue. And the idea here is pretty much this GIF, which is a bit slow, but it's showing us the same thing for the T2 image. We're starting with the lesion, forming midline alignment. We're extracting the lesion signal. We're extracting from the mirror image the healthy signal. We're smoothing the borders a little bit to make it fit the actual anatomy a little bit better. And then we're just implanting this image into the image where we reduce it, and we end up with this nice little healthy data. This was the idea in antimorphic normalization in 2018, and Solodkin came up with this virtual brain transplant idea to enable a free surfer and enable first approaches of simulation with TVB for tumor and for stroke patients. The other aspect is in the tractography, where we said the pathological anatomically constrained tractography, where we want to pay attention to the lesion in those tracts that we want to extract. And the classic approach here, which I will continuously afterwards call the baseline approach, is to, as I mentioned in the beginning, define the whole lesion as pathological tissue. And what this means in terms of anatomically constrained tractography is that when we are building these tractography images, we're starting at a seed regions, usually randomly distributed, and we're just following the probability distributions along these tract images that we extracted. And once we hit, for example, cerebrospinal fluid, we stop. So it is anatomically constrained. Once we hit gray matter, we stop, because the idea is might be following just white matter tracks. So here, if we introduce the lesion mask as pathological tissue, as soon as the tract enters the mask, we stop. Pretty much having a lower threshold of all traces that start and end in this lesion are being disrupted and are no longer present in our connector, which is what we see here. We're capturing everything that goes around. It's a little bit difficult to see this because it's a screenshot from a 3D image. So everything above the lesion we're still following, but nothing goes through the red mask of the lesion itself. And the last aspect that I mentioned was the containerization itself. So we make this pipeline available as a Docker container. The idea behind containerization is that it is a self-contained software unit, so there's no need to actually install anything to keep track of versioning, etc. All you need to do is to have Docker or Singularity in your host system, and then you can instantly apply the container, which significantly improves reproducibility, right? If the only installation that you have to perform is to actually pull the existing image, here the example of the TVB run to run TVB simulations from the virtual brain on Docker Hub, it's just Docker pull the image, and then you're ready to actually perform the simulations inside of Docker. That is a very good approach to enable really reproducibility and comparability across centers, etc. So what did we and how did we approach this in our pipeline? We talked about what the different steps are for correction, how the HCP pipeline is the gold standard of what we want to end up with, but also how the data does not fulfill the requirements to really run the full human connectome processing pipeline. So our idea is that starting with bits, which as the brain image data structure standard, it's the only requirement that we have in terms of the formatting of the data in the naming of the data files, the availability of the metadata, etc., the structure of the data. We require a T1 image, a high resolution T1 image, a T2 image, ideally high resolution, but it's able to handle flare as well, and the corresponding lesion mask. As I mentioned before, this is still the gold standard here is still manual segmentation, but this needs to be provided. There are different ways of segmenting this, right? A lot of the times we do this in flare, but also in combinations of T1 and T2 images, etc. So the only requirement here is to, when this lesion mask is provided, to actually supply the space in which this was segmented. If it was only done in flare, then we have to provide flare, or if it was done in T1, it has to be provided in T1, because based on this, we're starting the very first step of our structural processing pipeline, which is the cost mask creation. So as we mentioned in the cost function masking, we need the mask to exclude the lesion signal, and we are registering all over the place, all the different modalities that we have iteratively. So in the first step, we're taking this lesion mask and we're creating those masks in the different spaces by registering, already using this mask, the original image that it is based on, like I said, flare or T1, and registering it to the different spaces, even to MNI, because this is needed as well. And once we have this for T1 and the corresponding T2 image, we're performing the enantiomorphic brain correction, the virtual brain transplant, to have an approximately healthy brain for the specific subject. And with this now healthy brain, we're actually able to run the HCP structural processing pipeline. So the three steps that we've seen before, the pre-free surfer, free surfer, and post-free surfer, we're actually running this pipeline, while at the same time, each time we're actually performing a registration step, we're using cost function masking to really make sure that the impact of the lesion is as minimal as possible. So while we're using the scripts from the human connectome processing pipeline, all of those are just to cover the fact if we have the lesion and we need to perform these adjustments. All of this is done manually based on the prior steps. So if it finds the created folder for the lesion mask, it knows automatically it has to perform cost function masking, so it automatically applies it. If this has not been done because no lesion mask was provided, for example, in the case of healthy controls, it also sees this automatically and does not perform it. So then we're pretty much back to the standard human connectome processing pipeline, because we're talking about now healthy brains, and this is the gold standard that we want to do. At the end, we are running our own parcellation mapping. So if we want to really have this individual parcellation and subject space, we created a parcellation algorithm here that is based on the surface that extracted on the triangulations, and then applies the HCP MMP1, which is the Glasser atlas that is also developed within the Human Connectome project. Applies this to the individual subject space rather than on volume-based templates, such as, for example, in the MNI space, registering simply to the subject T1, which is significantly less accurate. So we end up with a parcellation image really in subject space that we've seen before, where we see all the different regions perfectly recreating the cortical ribbon of the subject. We're ending up with processed images, so the T1 and T2 image fully extracted, normalized, distortion corrected, etc. In subject T1 space. And we're ending up with a lesion properties, such as the load, as we've seen in the plot before, the load for each region of the lesion on that region of interest on that RI. And based on those images, the parcellation image and the processed image, we can go and run the next processing steps, where we now also include diffusion-weighted images. And the reason why we are not using the HCP diffusion pipeline here is, again, the clinical context limiting the data acquisition. So it is very rare to see stroke data that has, for example, reverse phase encoding, which is required to perform high quality distortion correction, which means that the same data is required, for example, ATPs, anterior to posterior, and then again posterior to anterior, really enabling us to have these scanner-induced distortions from both directions. The idea is then, if we are applying the distortion correction here, we can even them out and end up with the actual ground truth in the middle. Rather, we have just single shell, single phase encoding direction, so significantly less data and of less quality. So the HCP pipeline doesn't work. So we build our own based on Matrix 3. We're running standard eddy current, motion correction, etc. in the pre-processing, running population-based intensity normalization. In the next step, we're running this tissue segmentation because we want to end up with the cortical ribbon and the white matter, because we want to perform the anatomically constrained tractography. This is also where we introduce the pathological tissue with the lesion mask that we created here. Now in diffusion space, we're computing the response function of the FODs that we want to have to then perform anatomically constrained tractography, those tracked images that we have, and then use SIFT in combination with the parcellation image to pretty much just count all the tracks from one region to another region, so that we can end up with our structural connectome. And the same, or the similar approach we also have for the functional processing, where we actually able to use the human connectome project functional volume processing pipeline. And then we just add an additional T1 registration and then compute our functional connectome by extracting the average time series for each of those regions, correlating them, and getting our functional connectome. Yeah, exactly. This is actually just the result that we end up, right? We start with our anatomical image, raw already, still including skull, etc. And we're performing the structural processing pipeline, and we end up with our parcellation image in this image in subject T1 space in high resolution on individual parcellation. The same as we're starting diffusion imaging, we have the tractography, we end up with our connectome. And for the fMRI, even though we're starting with this very low quality fMRI data that we have, we're seeing with these missing regions, we are able to create functional connectomes. And here we actually see those white rows and columns are the regions that have been cut off because of the missing field of view, right? This go from the visual cortex, temporal cortex, etc. even parts of the frontal lobe here are cut off. So, but we're still able to create a functional connectome so that when we want to run simulations, we have sufficient data to validate our simulation at the end. The last step was the containerization, the last aspect for really enabling the reproducibility and the sharing of this. We created Docker and converted it to Singularity containers so that it can be run on high performance computing clusters and supercomputers. And the actual call is very, very simple because everything is done automatically and it assumes the bit standard. So what we just want to have is typically in a container call, we're just mounting the data that we have into a data directory inside of our container. We define the subject ID. This following pretty much bit standard. So this is the study folder containing all of our subjects, and then via the subject ID we are selecting the corresponding directory, and then we're defining in just a string the different steps that we want to perform. Here, for example, we already processed structural data, we already pre-processed the functional data, and in the last step we're just running the functional processing to create the connectome. So we already have the pre-processed data. Now we're applying the parcellation, getting the average time series, and computing the correlation. And we are still including lesion here to make sure that the algorithm actually pays attention to all the corresponding corrections that have to be performed in lesion. If we just look at the help function, if we don't provide any input, this is what we see. The subject ID that we require, the path that is equivalent to the study folder, and then we have a bunch of parameters that we can provide based on the steps that we want to perform. Right? We can just say full, which means run all the processing steps together. Based on the structural, just run the structural, all of them, just run the pre-FS step, etc. And based on the different steps that we want to perform, we might be able to provide the atlas, for example, just to say, okay, now we don't want to use the HCP and MP1, but we want to use a different atlas. And then we can provide the atlas string here as well. Currently, automatically implemented is just HCP and MP1, but this can be extended in the future as well. So what we end up with if we actually run this pipeline is we have our input data that we have fully processed. So we have our structured connectome, we have our functional connectome, plus we have this lesion load parameter table that we can then all combine into the virtual brain model, which is the final goal that where we want to end up, right? So how did we validate it to make sure that this is actually robust and it works and it is accurate? So one of the problems in validating such pipelines is that how do we want to infer the impact of an artifact when there is no way of controlling for the artifact itself? Because in the data we have the lesion. There's no way of not having the lesion and comparing, because this is the case, right? That's pretty trivial. So the idea that we had is to create a pipeline to call artificial lesion embedding, where in our subject population that we have in our study, we're actually taking a healthy control, which are age appropriate to our patient population, etc. So they're all matched, and take lesion signal that we have from an actual patient and integrate this lesion into the healthy subject, so that we can create artificial stroke patients that we can then process, and where we also have the ground truth, the healthy brain actually available, so we are able to compare how close we get to the ground truth if we are performing our corrections versus we're not performing our corrections. So to visualize this a little bit, we're starting with our healthy control subject, and we're looking at the region here. It's a nice, little pretty cortical folding. Everything looks good. And then this is how it looks afterwards, after we implemented the lesion. We can clearly see the lesion here, even following the anatomy of the subject at hand. And we see this in this point here as well, and in the cortical and subcortical areas. And here we see the original patient where we extracted the lesion. We see it is pretty similar to what we have in our artificial patient, but it is not exactly the same. And this is because in our pipeline we are performing some thresholding and some smoothing to make it fit in the actual individual anatomy that we have here. Classically, what has been done for example, training segmentation algorithms is in the early stages just to introduce a black blob, which is pretty much just a hole in the brain, which does not really represent actual stroke data really well. And then the next step, the next iteration of what people did was pretty much just taking the lesion and implementing it based on a published data set of lesions, which led to more realistic looking data, but still we had mismatch with the anatomy of the actual subject. And with our data now, we have stroke lesions where it is basically impossible to differentiate between the artificial patient and the actual patient, unless we're talking about having actual multimodal data available. But based on this T1, it is pretty difficult to differentiate just on the T1 images, which was the goal for us, so that we can have a robust approximation for our validation approach. So what we did is we used the ground truth that we have, the healthy subject. We used our artificial lesion approach and implemented the lesion signal, and then we performed 3 different processing pipelines. We performed the normal pipeline for the healthy control. We didn't need to do any lesion correction, so we're pretty much just running the HCP pipeline with our diffusion weighted pipeline, etc. So this is the whole pipeline up to the point of the parcellation, the whole structural pipeline without any lesion correction. Then we ran the same for the actual lesion patient, so we could see how the lesion impacts the processing without our corrections, and we ran it with the lesion correction to get results that would represent actually processing lesion patient data. And then with those 3 parcellations that we extracted, we computed agreement measures and differences between the ground truth and the not corrected parcellation, and the ground truth and the actual corrected parcellation. And then we compared the results that we had here. And then we used this parcellation because we wanted to know how those differences might affect downstream processing and even TVB simulation. We used those 3 different parcellations that we had, all with the actual healthy diffusion weighted imaging data that we had, and ran the diffusion pipeline, created structural connectomes, and then did the same thing by comparing the difference between the structural connectomes of the ground truth and the not corrected version and our corrected version, and then perform statistics to see how different they were. And if you look at local metrics that we extracted, for example, if we look at volume differences for a specific region of interest, so how just the volume of that specific region, for those regions that were affected by the lesion, we see a significant difference if we corrected versus not corrected. So we see that was a one-sided t-test, so we see that the volume difference between ground truth and corrected or not corrected is significantly larger in the not corrected case than when applying our correction. The same thing goes for all the volume differences. It's just not very visible here. We're still significant if we're looking at all regions, not just those affected by the lesion. And then we looked at the center of gravity for each of those regions of interest and compared how far those center of gravities were from the center of gravity in the ground truth case. And we also see that the not corrected case, the spatial distance between the center of gravity of those regions was significantly larger in the not corrected case, pretty much showing a larger distortion of the region when it came to not performing our correction versus performing all the corrections of our pipeline. For the lesion ROIs, this was not the case for all regions, but this is simply due to the fact that we're talking about a large number of regions which were just not affected by regions, so there was no real difference to expect. To visualize this a little bit, here we see in black, this is the cortical ribbon extracted, overlaid with the region of interest here in yellow for the ground truth. And then this is how it looked for the not corrected. It's the same region from overlaid with the same cortical ribbon of the ground truth, how it looked for the not corrected case. And we see a significant distortion and movement of the extracted region of interest away from where it actually was compared to the ground truth. So if we're now looking at the spatial differences, for example, in the center of gravity, we're moving pretty much outside of the actual cortical ribbon. We're now no longer in the actual inside of the actual brain. So the difference towards the ground truth actually is pretty large. This holds through from the local level to the global level. Even if we're looking at the structure connectome, we're performing the comparison here. If we're looking at the correlation of the connectome, the structure connectomes with the ground truth, we see a significantly, barely significant reduction in correlation if we don't perform the correction as opposed to if we perform our corrections. Not significant for the geodesic distances between the structure connectomes. But if we look at the connectivity profiles of the actually affected regions of interest, we see again a significant difference if we correlate those connectivity profiles, whether if we could perform correction and we do not perform the correction. So we see a significant drop in correlation there. So the actual connectivity profile that we see in those affected regions is very large. The difference is very large to the ground truth if we do not perform our corrections. So this gives us a fairly robust validation of the pipeline and of the approximations and corrections that we perform. We also validated some of the actual processing steps that we want to look at, because before we ran the validation and we just saw with the significant results, we did the same thing and we found that our data was significantly different from the ground truth for both cases, whether we performed the correction or not. So we looked at all the different steps that might cause this, even because we hypothesized that performing our corrections would significantly improve, which we just showed. But initially this was not the case, so we looked at the actual processing step that we perform. And one of the first processing steps where we saw that there is a significant impact in applying this to, for example, the patient group, but not to the control group, so having an asymmetrical application of this, is the actual cost function masking itself. And to validate this is, we took the same brain extracted T1 image for different patients, from 450 HCP patient, for 60 apnea patient, etc. And we registered this to the MNI space, one time just normal registration without cost function masking, and the other time with cost function masking. So we just applied the whole brain. So the idea is that this should not be different, right? If we're looking at what this actually means, is it says register T1 image to MNI space for the not cost function masking case, and for the cost function masking case, it says register T1 image to MNI space, but only use everything in the brain, because the mask itself is the full brain. So hypothetically, there should be no difference. We registered this to MNI space, we performed segmentation, we applied, because now we're in MNI space, we applied the HCP MMP1 atlas, and then for the ethnic case, we also created functional connectomes to see how really all across these different processing steps, these downstream processing steps, how does the simple fact of whether or not we apply a mask, even though everything is included in this mask, impacts the actual processing steps. We then looked at just global agreement measures to see whether or not there's a volume in the extracted cortical ribbon, there's a difference in the global correlation between the functional connectivity matrices, but we also looked at local matrices, again, seeing whether or not there's a volume difference for each of the regions of interest, etc. And at the end, we also, because I wanted to see how all these downstream processing and analysis steps are impacted, we also performed classification for the ATNI data set that we had. So we classified healthy controlled versus MCI and Alzheimer patients, and then tried different groupings according to whether or not we applied cost function masking or not. And what we ended up with is that if we're looking at agreement measures, such as the DIE score or the Jakar score, which pretty much measures how much an extracted image or a 3D shape, for example, in our case, the cortical ribbon, how large the overlap is, how much they agree. We see that for the different patients group that we had, or the different subjects group, HCP is 445 HCP subject, healthy controls or patients from the ATNI data set. We see that while we have a rather high DIE score, we still see an impact of applying masking, even though hypothetically or theoretically there should be none, to the registration. And then the same goes for the volume difference. So we see a difference of up to minus 0.08, which is equal to a reduced cortical volume of 8% when comparing cost function masking versus no cost function masking in the extracted cortical ribbon. If we break this down per region, we also see this the differences in the volume differences, again from 0 to 10% for the different regions, the 368 CPM and P1 regions. And this is true for FSL, this is true for ANS, and this is also true for SPM, which we didn't include here, but we also tested. So this is not a question of implementation in the registration, but this is rather inherent in the method itself. Somebody's listening to music, that would be nice if you could mute yourself. So this is what we see even on the local level. If we break it down a little bit more, if we're looking at averages across the whole population, we see on the left side ANS and on the right side FSL. So it's again not based on the actual software. We see quite a pattern emerging of the regions that are mostly impacted from whether or not we apply cost function masking. So the chi-score goes down to 0.8 here, which represents a poor matching of the extracted regions between performing masking and performing no masking. And in the same way, we see this here for the classification that we perform, the random forest classification for Alzheimer patients versus healthy control. If we just perform this classification and we did not mask either of the data, and we just classifying based on the regional volumes, we get the regional volumes, and we get a classification accuracy of 67%. And then if we map the feature importance, so the top 10% feature that drive the classification accuracy, the classification itself, we see this pattern here, these regions as the main contributor to the differentiation between the classes. If we do the same thing and both of them were masked, so we applied cost function masking to patients and to healthy controls, we see roughly the same accuracy, and those are averaged over 1,000 runs. And we see a slightly different pattern. The temporal lobe here is no longer that important, but here we see roughly the same pattern emerging in terms of what is driving the classification. Now, if we apply cost function masking to either the patient or the healthy control, but not the other, so we are applying it asymmetrically, we see a totally different pattern. First, we see a significantly improved classification accuracy. So all of a sudden, we see 87% accuracy in classifying healthy control versus Alzheimer patient. But we see that the regions that are now popping up as being the main drivers of the classification no longer match what we've seen with the actual, where we actually don't have the impact of the cost function masking. And most interestingly, we see that if we perform classification within the same group, so we're talking within patients or within healthy controls, but between masking or no masking, again, we see this 85% accuracy, and we see pretty much the exact same pattern emerging, while the only difference is that actually masking was performed. So we can see that if we asymmetrically apply cost function masking, for example, for our stroke patients, but not for our healthy control, we might artificially inflate those group differences just because the model, and here in our case, this is the machine learning model, is encoding information about the masking rather than about the actual disease case, which represented in a different pattern and also significantly less accuracy. So what we need to do is we need to pay attention for even these established processing algorithms, that they can impact the final inference that we want to take from our model, in this case here now machine learning, but we can also show this in a second for actual TVB. And the other part is, as I said before, the way we integrated the lesion in the tractography was we set everything to 0. So we applied the mask as pathological tissue, and we said everything that's hitting a voxel that is part of the mask will no longer be counted. But we see that this is not necessarily the case, and this is a very conservative measure of implementing this lesion information. And we see here for multiple sclerosis lesion in this publication, which is really nice from 2020, where we have this lesion being represented inside of the gray matter, as we can see here. And again, we have this dark spot of the lesion that is represented as compared to the rather bright white matter surrounding it, but also we see that this is not all the same intensity values, right? So we have gradients inside of the lesion as well. As you've seen with the intensity distribution, those are rather flat. We have every value pretty much being represented in the lesion itself. And they looked at the actual probabilistic directions inside of those voxels and found that while inside of the lesion, in the darkest spot here, we see probabilistic patterns that are very similar to what we see in CSF outside here, which represents a feasible and viable barrier for anatomically constrained tractography. We see that in the only slightly grayer regions, which are still part of the lesion itself, we see patterns that are still somewhat representing what we see in the white matter. So the idea of setting all of this to 0 might be an overkill in term, and we might lose information about the actual lesion that we have, especially if we consider what we've seen at the beginning, where we have longitudinally, we have this dynamic of the lesion, and most likely all these gray areas are in the subacute and in the chronic state, no longer represented this way, and they're no longer part of the actual lesion itself. So setting this to 0 might artificially, again, inflate differences that we have because we're excluding information which is actually still there. And we see this if we perform tractography with including the lesion mask here, as we said, so we don't keep any traces that go through here. And if we do not do this, so our pipeline enabled us to select the embedding mode of the lesion. And we could also say, do not include the lesion mask as pathological tissue, but rather just track everything that goes through as if it was healthy signal. And we see that actually the way these traces go, especially with the hole here in the middle, is very representative of what we also see in the lesion. So we see that the center of the lesion here, the very dark spot, is actually not tracking any tracks, but rather we have tracks going around and trying to circumvent this that are still viable and still valid. If we think about the slide before, there are still tracks, and the probability is still not 0 that these tracks might actually be there and then also recover over time. So figuring out a way of integrating this into tractography and also then into the functional and structural connectomes and into the TVB models will enable us to get, even for the acute data, a more accurate representation, and then maybe enable us to have more of an accurate representation of the recovery of those regions, and with the regions, the patient itself and the symptoms. So if we now look at the simulations, what has been done so far, there's some great papers in 2015, 2016, Falkenau, using stroke data and using what I call the baseline approach. So what they did is they included the lesion mask as pathological tissue in the tractography, and they also set all the structural connectivity weights to 0 and ran the simulations. And we see the simulated and the empirical time series here, and we see the simulated and the empirical functional connectivity matrix. And then they looked at the different patterns of global coupling and conduction speed to compare healthy controls and stroke patients itself. So what are the steps that we are now taking for simulations? This is the baseline where we're starting. We now have the pipeline that easily creates us TVB ready data, and we can even select the different ways of how we want to integrate the lesions. So we are now, or we already did, run multiple processing runs of those integration. So we include the lesion mask in the anatomically constrained tractography, which is the baseline, which is the other papers that we've seen in the literature. But we also did it without, as we've seen in the plot before, where you can still have the tracks go through the lesion. And then we developed multiple ways of integrating the information that we have of the lesion into the actual model, so into the structural connectomes that is the base of our TVB model. So again, as the baseline, we're just setting all the weights to 0. Then we are not performing any specific lesion embedding at all. So we're taking the structural connectome as we get it, and also we adjust the lesion where we have the affected region of interest with the parameter that we extracted, right? Where we have this this load on the region of interest, which is between 0 and 1, and then we update the weights for the connectivity matrix based on this parameter. And what we end up with first is the different ways of integrating lesion and ACT. So we have a structure connectome without integrating lesion as in ACT. We have the structure connectome after integrating lesion in the ACT. And if we look at the difference, we actually see a strong difference in the structure connectome. Down here, we see the actual regions of interest that were affected. So we're just talking about right hemisphere and subcortical areas that are affected, which explains the large differences here. But we already also see huge differences in the left hemisphere, even though they are not directly impacted by the regions. So this is, if we perform lesion-based ACT, this is information that we are losing about the other hemisphere, because if we're talking about subcortical areas that are affected, there are still areas in the other hemisphere where we have impacts. And this is just the ribbon of the actual lesion mass that has been applied here. So you see a lot of areas in the right hemisphere that has been affected, and here just to have this mapping to the structure connectome matrix. So what does this mean? How do we now, based on those two different approaches that we have, how do we integrate it into the structural connectome into TVB? We have the lesion integration in ACT, we have no lesion integration in ACT, and then we have the three different approaches, whether or not we just take the raw weights as the model for our TVB model, raw here being not adjusted for the lesion. It's not actually raw, right? Those are normalized and thresholded, etc. Some standard stuff to perform for structure connectivity matrix to integrate them in TVB. Then we have the way where we set all the weights to 0 for the lesion affected region of interest. And then we have the way where we update the weights based on the load that we have. And we see the largest difference here in the subcortical areas down here. If we do not perform any specific integration, if we set them to 0, and if we have the load update the actual lesion. And this is even after integrating lesion into the ACT processing. If we do not perform the ACT processing, we see a similar pattern. So the question is, how much is this a trade-off by losing information? If we include lesion in the ACT, how much can we reverse that loss of information if we rather just include it but upload the weights, update the weights based on the load, etc. So we're talking about the same subject and different ways of integrating this. So the next step would be, how do we validate this, that our TVB model is accurately representing this? Because even those are slightly differences, and we see here those are minor differences that still show up. We see a significant impact on the actual TVB simulations that we have. What we see here is just some very basic grid search for the same subject for those 6 different data types, which we have all being pretty much the same structure connector. We started from the same data. We just took different decisions on the processing, on the integration itself. But then if we look at the validation, so correlating the time series that we simulated here with, for example, the combination of the global coupling and how do the correlation looks in this grid search with the empirical FC matrix that we have, the empirical functional MRI data, and we see that we have a significantly different pattern. So the interpretation of what the combination of conduction speed with global coupling actually means significantly depends on our decisions of the processing and our lesion integration, similar to what we see, for example, if we're talking about the cost function masking, we see at the end that downstream analysis might be significantly affected by processing decisions that we made. So the question is, our model, if we here our baseline model where we have the integration in anatomically and ACT, and we have the integration as just 0 weights, what does this mean? What does these high accuracy for this conduction speed and global coupling combination mean for the model in terms of, is it encapsulating or is it encoding disease information, or is it just encoding processing information if we see the pretty much exact same data representing, showing totally different, significantly different profiles for the same parameter combination, but for different processing decisions. And with this, just a quick summary. What we've seen with the lesion to DBB is we have a containerized, fully automated pipeline that first is the first implementation of this virtual brain transplant out there, but also is able to handle stroke lesion data, these kind of artifacts, but also low quality data, as for example, present in the clinical context. And then enables us to really, for example, use TVB to study underlying mechanisms and to try to get our model to be more driven by the disease rather than by processing decisions itself, and really validate those. And we just see that the processing itself and through our validation framework creates significantly improved performance in terms of accurately reproducing the ground truth for the artificial lesion embedding that we tested, and also for the cost function MASCI, where we could show that these artificial processing based inflations of group differences can be avoided and have to be paid attention to. And I think with this is that's pretty much it. I didn't check the chat in between if there were already some questions. I don't know, but happy to answer any questions now. There still are some or somebody. I guess that means there are no questions. Cool. Yes, there was a very clear talk. Just going to decide to look at it this way. Julie, I cannot, you're welcome. I cannot stop the recording. It says an organizer.